Hi everyone, this is Morgan Berger. I'm here for discussion number 2. I decided to take the components of the cell and use colored pencils to draw them individually and describe them one by one, and then at the end put them all together into one cell to show you. So first, in the drawing I have here, we can see the plasma membrane, which is represented by the yellow. The cell membrane provides the cell with shape and protection and allows for communication between other cells. The membrane is semi-permeable and selective permeable, which means that some things may enter the cell and leave the cells while others are unable to. Also in this drawing, in the red, you can see the cytosol. This is the fluid that everything floats in and is mostly made up of water. It is sometimes referred to as the intracellular fluid. In this next drawing, I have the nucleus in green. This is the control center of the cell, which contains the DNA. The black dot in the center is the nucleolus, which creates the ribosomes, which I will discuss later. This black circle is the nuclear envelope, and it is a bilayer that surrounds the nucleus. Next, we have the rough endoplasmic reticulum, or ER for short. The rough ER contains ribosomes, which can be seen here and are shaded in a little bit of the darker blue. Ribosomes are in charge of the peptide and protein synthesis. Here we have the smooth ER in the pink. This does not contain ribosomes, but it detoxifies the cytoplasm. Endoplasmic reticulums have channels called cisternae. Here in the dark green, we have the Golgi complex. This completes peptide and protein synthesis. And also seen in this picture, you can see the Golgi vesicles, which I will show here a little bit closer up, and also dark green. These finish the proteins and are transported through the cytoplasm. Here in the purple, we have the mitochondria, which is better known as the powerhouse to the cell. This is where ATP is synthesized. These blue circles are lysosomes. They are special Golgi vesicles that do not break down organelles. They are small pockets that have digestive enzymes. Here in the brown, we have peroxisomes, which are created in the ER and detoxify cells by using a hydrogen peroxide to break down the particles, and they can be seen here. Finally, we have the centrioles in orange. These contain microtubules, which are proteins used in cell division. And here we have one whole animal cell with all of the organelles. So here in the light green, we have the nucleus. The nucleolus here is the black dot. The black circle around is the nuclear envelope. Here we have the rough ER and the ribosomes along the edges, the smooth ER. We have the Golgi complex and the Golgi vesicles. We have the mitochondria up here, the lysosomes, the peroxisomes, the centrioles, and then around here in the yellow is the plasma membrane, and of course the cytosol. Everything is floating in the red.